So welcome back to another video, and in this video we are going to talk about a very important concept and a very interesting one, which is the role of p53 gene in the formation of cancer. So p53 is a transcription factor, it is helping other genes to get transcribed. It is activating the transcription of other genes. And this P stands for protein, and this 53, this is the molecular weight of that protein, that is 53 kilodaltons. So that is why the protein is altogether called p53, and the gene that is encoding this protein is called TP53. So this T stands for tumor, and p53 is, as usual, protein 53. So tumor protein 53 gene is encoding the protein which is a transcription factor, which is called p53. Now, since we have the basics down, now let's talk a little bit about the signaling of p53, how it works. In normal condition, when a cell is in normal condition, there is this continuous expression of p53 from the TP53 gene. So this expression is in a basal level, and there is another protein which is called MDM2, which checks the level of p53. What it does, it binds to p53. This is p53 and this is MDM2. So MDM2 binds to p53 and makes it degrade. So the p53 in normal cell, p53 is very low. Levels are very low because MDM2 is degrading it. Now let's say there is a damage in the DNA. It can be due to UV radiations or conditions like hypoxia, which is very low oxygen concentration. Now, these conditions may lead to the damage of the DNA. Now, this damage in the DNA is sensed by two proteins, which is ATM and ATR. These proteins are actually serine kinases. So these ATM and ATR, these proteins, they phosphorylate other proteins at certain serine residues. Now ATM and ATR, what they do, they phosphorylate p53 and MDM2. Now, since p53 and MDM2, these are phosphorylated, they are no more bound together and is freed from one another. Next, the free p53, what it does, it activates the transcription of a gene which is called WAF1. So p53 is activating the gene which is called WAF1. Like I said, p53 is a transcription factor. It's activating the transcription of another gene which is WAF1. Now WAF1, the protein that it encodes, is called p21. Again, the same thing. It is having a molecular weight of 21 kilodaltons. So that is why p21. Now what happens in a normal cell? There is this transition from G1 phase to the S phase. This G1 phase is called the gap1 phase in the cell cycle, and this S phase is the phase where DNA is replicated. So when there is a signal for cell division signal, the cell moves from G1 to the S phase, so that the DNA that is present, that can get replicated and that replicated DNA can move into the subsequent daughter cells. So this process is mediated by this complex, which is cyclin D CDK4 complex. These CDKs are actually cyclin dependent kinases. So these CDKs, they bind to this cyclins, and then only they can perform their kinase activity, that is transferring a phosphoryl group. Now in normal cell, this is the thing. So this is happening in a normal cell. When there is a cell division signal, the cell transits from G1 to S. Now when there is damage in the DNA, that DNA, if it is replicated and moves into the daughter cells, that might lead to complications like cancer, because certain genes may be mutated because of the damage. So this whole p53 signaling, what it does, once p21 is encoded or translated, it inhibits this cyclin D CDK4 activity. So cyclin D CDK4 activity is inhibited, and the cell cannot transit from the gap1 to the synthesis or S phase, and the DNA is not replicated. So this is done because in a cell with a damaged DNA, this buys the cell some time to repair the DNA. So if the DNA is mutated, for example, mutated in the G1 phase, this inhibition sort of gets the cell some time to repair this mutation, repair this damaged DNA. Okay, so p53 is not directly involved, but it is helping the encoding of p21, so it is somehow indirectly involved in repressing the cell cycle. And since it is repressing the cell cycle, that is, it is not letting the cell to proliferate or to grow, that is why it is called a tumor suppressor gene. That is, it is helping a cell not to form tumors, not to grow abruptly by inhibiting the cell cycle, and it is suppressing the tumor formation. That is why it is called tumor suppressor genes. Now we know that the human DNA is diploid, right? There are two copies of every gene. Let's say this and this. These two are homologous chromosomes, and these two loci on the two homologous chromosomes are having p53. The gene TP53. Now, say we have UV radiation, and one of the TP53 gene is mutated. When this is the condition, there won't be any problem, because I have another TP53 gene which can produce a functional p53. Although the secretion or the translation would be in a lower amount, but the p53 functional gene would be encoded. Now, if both of these genes are UV radiated and is having mutated, then there would be a problem, because I do not have a functional p53 gene. Now, this is why a recessive mutation on both the TP53 genes will turn a cell into a cancerous one. But if only one of the TP53 gene is having a loss of function, that's quite fine, because the other TP53 gene is all right, and it can encode a functional p53 protein, and that can do all the function. So a recessive mutation in in both of the genes, or in both of the loci of the chromosomes, the homologous chromosome, is required for the formation of a tumor or a cancer. So that was all about the function and the pathway by which p53 gene is regulated, and it is regulating the cell cycle and the formation or repression of cancer.